I would like to introduce our next speakers. So it's Bette, Korber and Will Fischer from the Los Alamos National Laboratory, and they will be presenting a resource for SARS-CoV-2 mutation tracking and analysis, the LANL GISAID COVID pipeline. So Bette and Will, I'll hand it over to you.
Will, I would just like to open this for one moment and talk about a little bit about where this came from. It's a very specific sort of set of analysis tools that came about because a lot of people in March and April who are working on the current vaccines that are being deployed and tested, not deployed yet, but tested in the different clinical trials, we're interested in trying to figure out what would be the best set of variant reagents to have available for looking at antibody responses in particular. So this is a very focused set of tools that was created to provide a little bit of interpretation to the massive amount of data that's pouring into GISAID to address that question. What are the variants that are arising? Are any of them relevant? And some of them are likely very relevant to be put into the panels for looking at cross-reactivity of antibodies. So that's sort of the framework of how this was developed, and that's the focus of the questions. And Will's going to open the talk, and then I'm going to finish it up with some demonstrations.
Super, thanks Bette for that introduction, and thanks Nina for likewise. So the IEDB has invited the Los Alamos HIV group to present at this workshop for the past several years, and we appreciate the opportunity to bring our specific pathogen-focused viewpoint, data, and tools before a wide slice of the immunological community. So for decades now, the HIV database at Los Alamos National Laboratory, LANL, that's LANL as I'll say it, has curated HIV1 sequence and immunology data. And our colleagues Brian and Elizabeth will present details of that work tomorrow. This morning, we're going to talk briefly about some highlights of this year's new efforts. So this year, great year. We have a new specific pathogen, and we're using our virological, our immunological, and bioinformatics experience against a new devastating scourge. Among the surprises of this year was the absolute flood of viral sequence data and the extraordinary response of the research community. And like many other groups with relevant expertise, GISAID and the LANL HIV group rapidly deployed resources to respond to the pandemic. Now, the full name of GISAID is the Global Initiative for Sharing All Influenza Data. And that is a pretty nice description of their core mission. But they began collecting and sharing SARS-CoV-2, COVID-19 genome data almost immediately after the first genomes were available, and rapidly they became the go-to site for these genomes. At LANL, our experience with HIV and other pathogens puts us in a sort of state of active but useful paranoia. So we set up what is essentially an alarm system for emerging mutations in this new virus. And now we're a national lab, we're part of the national security apparatus. So I need to put that into national security speak. That is to say, an early warning alarm system or a forward-looking threat assessment for mutational escape. And as Bette said, this is focused on the what we perceive are going to be the needs of the vaccine and therapeutic community. So our website uses some of the code base of the HIV databases, and as I said, you'll hear more about that tomorrow. But we have a slightly different approach. We're not serving sequence data from this database because that's GISAID's purview. We're using their data to as a baseline for analyses that we perform and results that we'll make available to the community. And we were focusing on specific analyses of related viral sequences and looking particularly for the emergence of mutations in coding sequences and putting those in their geographical and temporal contexts. And we're paying obviously special attention to the spike protein, which is the major focus of vaccine and therapeutic efforts because it's the main docking protein for the virus. So for the last 7 months, we've essentially every day downloaded the complete set of sequences from GISAID, and that's gone from a few hundred to a few thousand to tens of thousands, and now in last night's build there were 178,000 complete or partial SARS-CoV-2 genomes. And we do a set of quality filtering because not all the sequences are complete. Some of them have stretches of ends. People handle ambiguities in a variety of different ways, ambiguities or missing data. And our goal is to generate contiguous files with uniform high-quality sequence available throughout a particular region. We align these and make phylogenetic trees from them. Now, this particular slide shows details of the filter and the results from the filter back when there were was 1/5 of the current data. So periodically, we've had to revise the details of the pipeline to cope with simply the size of the data, but we revised it periodically, so the data build on the whole database still runs overnight. Now the sequences keep pouring in, hundreds per day, thousands per day. We partition them, as I said, into different genomic region files. Each one is complete and high quality across the whole region. The idea being that we are reducing the noise so that we can see the signal. And what this graph shows just the growth over time for each partition, with the current counts over there all the way at the right for each partition. And just to note, the complete on the bottom goes from absolute, the 5' cap on the end of the genome all the way to the poly A tail, and that's a minority of sequences. Most people start with primaries that are slightly within the genome, giving what we call near comp for near complete, 29,782 nucleotides. We also provide a set that has slightly more sequences in it that goes from the first start codon to the last stop codon, again because we're focusing primarily, but not exclusively, on coding sequence mutations. So if you one of the main concerns is the different viral lineages, and the way we detect emerging mutations is by increases in frequency of a viral lineage that bears particular mutations. So this figure from GISAID shows in the center a circular phylogenetic tree, and the background shading denotes branches that belong to particular lineages, or I'll also use the term clade interchangeably. On here, all the pinkish or peach-colored backgrounds share a set of mutations that distinguish them from the original outbreak strain. And the G form here is the ancestor of all the two-letter clades, all the GR, GH, GV, each of which is distinguished from the original the G clade by its parent by additional mutations. And it's important to note that the original forms here are represented by a tiny little slice at about 315 or 310 in a clock face coordinate, and everything else has a substantial number of additional mutations. Now, like all databases, there are issues of sampling bias that need to be addressed, and those are particularly acute here. So in this data set, 3/4 of the sequences come from just 4 countries, and this is not exactly proportional to case rates. Though it is true that the United Kingdom and the United States have a lot of cases. The sampling is not directly proportional to them, and particularly there are many other countries where there are very few sequences despite fairly high case rates. And what's more, sequencing efforts have been episodic, maybe even spasmodic in different locations, so that inferring temporal patterns, inferring the dynamics of mutational evolution requires very careful work, and Bette will talk about some of that very shortly. But there are some interesting patterns that are visible if you look country by country. So these are the top 20 countries in terms of sequence submission, and they're colored by clade with cumulative submission counts shown by date from January up through the day before yesterday, I think. And you can see a number of different things. So first overall, in most countries, the initial forms, the warmer colors over to the right of the graph, over to the left of the key at the bottom, where O is the original. In most countries, those stop increasing fairly early and are replaced by the cooler colors, the other clades that bear the G, that start with a G, that increase rapidly and differently in different locations. So, for example, you can see that in July, suddenly a progressively larger proportion of cases in Australia in the top row. Suddenly you get this big blue spike, which reflects the increasing predominance of the GR clade, which is being sampled intensively in Victoria. And you should also note that where the line becomes horizontal, it's because no new sequences are being added. These are cumulative graphs. So in places like China, what you're seeing is essentially a frozen frequency distribution because they haven't added any new sequences in the last few months. But what you see is the original early distribution. So there's another way of looking at that. And this is a tool directly on our website, where you get pie charts showing the frequencies of different mutational clusters by frequency, and each pie is sized by the number of sequences available. And so this is the current distribution. But I mentioned that we designed this as an early, and Bette conceived it as an early warning system. So if you look back to sequences at the beginning of March, you see here almost everything is mostly yellow, which is the form that corresponds to the original outbreak strain at this particular amino acid site in the spike protein. So we're looking here at the frequency of amino acids in one site 614, amino acid 614 in the spike protein. And the yellow form represents a D there, an aspartate, whereas other forms represent a G, a glycine there. So back in March, before March, as I said, it looked like this. And at late March, it looked like this, and you can see suddenly, wow, all the yellow is becoming a much lower proportion of the total. And that trend continues. This is the beginning of May, and that's the beginning of August, where the yellow is almost completely gone, except I believe down there in Malaysia, and a few other locations there are little bits of it showing. This sort of thing is exactly what we're looking for when we're talking about looking for fitness effects, for things that we need to watch out for in terms of the effect on the pandemic, the effect on virology. So, as I say, back in March, it was all yellow. Before March, later it's becoming progressively more blue. And so it became important. This is something we spent a lot of time trying to figure out what's going on. And this is a summary of work we presented as a preprint this spring and published in Cell this summer. And the increase in frequency of the mutant form, which you can see in the bottom of the graph, is where orange is the original and blue is the new form. The increase over time in multiple locations, multiple independent samplings from across the world, led us to propose experiments that were done by our colleagues, which confirmed that there are phenotypic differences in this mutation, which are associated with higher transmissibility. And we'll note, right, this form has taken over the world since then. Now, later work done by additional colleagues and other people has shown why this has happened. The key point is the effect of this mutation on the structural dynamics of the spike protein. So this particular mutation leads to greater exposure, more frequent exposure of the receptor binding domain in its active position. So it's really a beautifully simple explanation for the greater infectivity. Having the receptor binding domain up more of the time means it's more likely to bind the receptor. Although this makes it more infectious, it fortunately doesn't appear to make it more deadly, and it may in fact make it more vulnerable to neutralization by antibodies. So this is an example of what we wanted to do when we set up this resource in the first place, was to look out for things like this so that they could be evaluated in terms of their potential effect on therapeutics and vaccines. But the evolution never stops. So as long as this virus is out there, it will continue to be subject to natural selection and mutational pressure, and will continue to develop mutations, which will expand if they're provide a selective advantage. And so Bette is going to tell you about some of the next big things that we're watching out for. And I just mentioned Bette's led this work from the start. It's been her project. So it's been fun to be associated with it, and thank you for your attention.
Thanks, Phil. I'm going to go ahead and try to share my screen now. And I'm going to attempt to do some real-time analysis and show you how to use the database directly as we're doing this. And we're going to put up these slides on the database so you can go back to them. They're a little bit detailed right now. So this mutation at position 614 showed this incredibly rapid transition virtually everywhere we looked all over the globe. So we wanted to make tools that enabled looking all over the globe. And we're keeping an eye out to see if anything else is showing up like this. And we're trying to help Project Warp Speed and David Montefiore's lab for the analysis of vaccine products for neutralization know exactly which mutations they want to have in their panels for resistance. So the most common mutations that are up as of right now that are maybe of interest are spike S447N, and I'm going to use this one as our example. And these are always found in the D614G context. So that newer form of the virus, that newer clade, these things emerge from that newer clade. There's another mutation in spike that's coming up fairly regularly. And these two are both the most common now. 6.3% of the sampling is for S447N and about 8% for the other. And then this other mutation, N439, is the most common mutation that's in the receptor binding domain antibody sites. So it's of interest too, but we're going to just use this 477 as an example. And the first thing I want to show you is just something really primitive, but it's something I've been using and I thought it might be convenient for people. So this is just, if you're studying antibodies for SARS-CoV-2, we're updating this relatively slowly, but it's a spreadsheet. And what the spreadsheet has on it is it has every position in spike, and it has some of the key studies of antibody contact residues for COV2. So you just go through, here's the position numbers, and here are the variants, and here are the local variants in a region of 10 amino acids, and the most common ones are highlighted in yellow. And then these are different antibody binding sites, or known as escape mutations. Just within the spreadsheet, we're trying to make these into nicer database tools over the course of the next few months. But here you can see these are a lot of different antibodies in the receptor binding domain. These are contact residues that are highlighted. And so what you can do with this is you can look directly at the domain that it's in, the position that it's in, the frequencies of mutations in this site, and then sort of what's known about the site and its emergence of resistance from a few of the papers that have come out. And we're trying to update this on about a monthly time scale. But this isn't fancy. This is just something that I thought might be handy for people. So I wanted to point out that it was there. But meanwhile,
we're only seeing your slide. We're not seeing your spreadsheet.
Really, you're not seeing the spreadsheet? Are you seeing the database over here that I'm just moving?
No. No.
Oh, that's so interesting. Gosh, this may not work then.
You're just probably sharing the wrong window.
I'm sharing this. It's all in my one screen. So your screen sharing, and so you can only see my slide. Well, then you know what I'm going to do is I'm just going to talk you through the slides and not show you how to implement these things on the database, because we have examples from the slides. So if you can't see them, I think that'll work. So here is an example from the spreadsheet. This is site 477, the one that we really care about. And you can see the number of variants that are found in this site. So S is the most common in the original form. These are all of the variants, how common they are. This is how close it is to ACE2 and whether it impacts ACE2. And then this is if it's in antibody epitopes that are known for the literature. So that is available. You can also look at tables of variation in spike. I wonder if I take this off. Can you see now? The database?
We just see a white rectangle.
Shoot. Well, that's very strange. So you're not seeing my active database interface. I don't know why that is. At any rate, we create tables every day based off that alignment that Will showed you. And in those tables, we have all of the sites that are frequently mutating and everywhere they're found in the world. So if you go down to our tables list, you'll see that. You'll see the variation in all positions in spike, and you'll see every country that holds those variants and the frequency, and every city, every level of geographic information in GISAID. And why we think that's interesting is because if you're say have a site that's in the receptor binding domain and it's dominating your country, you might want to know about that as clinical trials go into your country with an antibody or a vaccine that's targeting that site. And you might want to know whether it's relevant to the vaccine response. So some of the things that aren't really dominant in GISAID, but just dominant in one geographic area, may be useful information for vaccine trials. We can track that really easily in these tables, and they're updated daily. And so 477 turns out that it's really the common variant in most places in Australia. And then using a tool called Analyze Align that we have, you can see how often variants are occurring in sites of interest. And if you have an epitope of interest, for example, whether it's continuous or discontinuous, you can see how much variation there is in that epitope. So we have two tools that search all of GISAID every day. And they look for geographic locations and change in frequencies of sites. And they pick up every single geographic location in GISAID where there's a change in frequency, whether it be up or down for a particular mutation. So site S44N, which has increased to 6.3% of GISAID, is really dominantly found in Australia. And the way you would read these isotonic regressions is the following. They have dates along the bottom, and every single little dot represents a day. So if a day was sampled a lot, it has a big dot, like Norway here, in this dot. And if it was only sampled a few times, it has a tiny dot. And this is the fraction of the mutation in that location. And it only shows the ones where there's a change. It could be either a change up or down. In the case of 477, it's always increasing. And you can do this again at the country level, which I show here, or at the state level, or at the city level. And you can see, given the GISAID data, how things are changing. So this particular site's going up in a lot of different countries. An important feature of this is looking for the probability of the mutation. So it's happening quite frequently in Australia. It's almost 100% now in Australia. And in France, it's going up. But in the United Kingdom, it's going up very slowly. And this is only 10% here, so it's not very high. So this is one of our tools to look for change in a mutation over time. And 477 is right at the edge of the receptor binding domain and actually in a couple of different antibody epitopes. So it's an interesting site. This is the other way we have of looking at changes in frequency over time. And this just samples when a mutation first enters a population and it's first detected. We look at its frequency at that first detection point in different countries. And then we look at the frequency of a paired frequency 2 weeks later. And we ask if there's been a change. And it could either, again, go up or down. And again, this site's always increasing. So as the blue bar gets larger, it means the mutation is showing up more and more in those countries. And it's highly significant. So these things go through all of GISAID. I'm sorry.
4-minute warning, sorry.
A 4-minute warning. Okay. So this site, as it turns out, is really dominant in Australia and a few places now in Europe is coming up. But the reason that it's such a large part of GISAID is because Australia is really heavily sampled during this period in the summer. And so here's now a tracking map, and you can track any mutation like this in any region of the world or globally. So here's the global map of this mutation. Here's the original form. The blue form is the new form. And you see this blue sort of surge of this form. That's because a lot of sequences came in from Australia, and it dominates the Australian epidemic. So knowing how this site would impact antibodies and vaccines is important for Australia. And it's also important for the parts of Europe where this is emerging. So it's a good one to have in a panel for testing. That's sort of the bottom line. You can break this down further and you can look at all of the different regions in Australia. You can see that it's really come up in Victoria and well sampled. It's come up in Sydney. It's come up in Queensland over time. So the original is orange, the mutation is blue, but it hasn't yet reached Western Australia. This is something I think that is useful in general for immunologists. This is the thing where you can put in, it's called analyze a line. You can put in a list of sites. They can be contiguous if you have like a T cell epitope that you're interested in. It can be used in any part of the protein you would like, or it can be discontinuous. So here are just 4 positions in spike that are mutating. And you can see how often you have mutations in these 4 sites. So this is the GD mutation that now G dominates the world. So it's 26,000 times sampled, whereas the original only 202 times sampled. This is actually from July to November. You can set a time window for this as well. So by July, there's hardly any of this D left. But you can also see the frequency of these other forms. So this V form 222 has been quite common since July. It's now 10,000 times. The S477 is quite common. And these are, again, really skewed by the sampling bias of the UK and Australia, where these things are heavily present. And they're very seldom found together. And this might well be recombination. The virus is recombining. We also have trees where you can look at any mutation you want to in its lineages of these common mutations. So we only have the common mutations. But here we're looking at just the one we're interested in right now, 477. And you can see that it's coming up not just in one clade. So here's this really huge Australian sample. It's all from one clade, all one lineage. But you can see that a different lineage is coming up in Europe. So all of the UK, Switzerland, and France sequences are from this lineage. One particular region of the UK has got a distinctive lineage, and Sweden's having it come up in yet a different lineage. So this particular mutation is coming up in different backbones in different places of the world, and yet it's still increasing in all of these places. So these are just tools for the way they would be relevant to an immunologist, I think, is if you're interested in a particular epitope and you want to know if that epitope is changing, if there's multiple mutations in that epitope, where they're frequent, where they're rare, then you can do this. And the other thing I just wanted to mention right now is something I'm really excited about. So we've been working with GISAID one of the things that's going to be happening with the big clinical trials for antibody therapeutics and for vaccines is that the FDA and other groups are requiring that they provide sequences for breakthroughs. And so hopefully what that means is when these vaccine studies get published over the next year, there may be thousands of sequences coming through that may have immune pressure on them given the particular trials. And it would be really nice to be able to have within GISAID information about the clinical trial that was conducted. And so far right now, they've just been keeping geographic location, health status, and time, the day of sampling. So they've decided to build and are in the process of building a very nice sort of clinical attributes to link to the sequences. So you can see if particular sequences have come through clinical trials. That's not open yet, but it's coming. So if you guys are involved with clinical trials, keep an eye out for that, because I think that's going to make the GISAID resource much more valuable in the end, and we'll be able to see if these trials are having resistance mutations emerge relative to the other sequences in the database, and if they're impacting a region beyond the clinical trial itself. So I'm very excited about that opportunity to be able to look at the evolutionary consequences of the large clinical trials as they go forward. And again, that's not there yet in GISAID, but it's coming. And if you guys are involved, please keep an eye out for it. And you can certainly write to me if you want to learn more about it, or the folks at GISAID. So that's basically our talk. And I'm sorry I couldn't show you in real time how to do these searches, but maybe the slides got you some idea of what's there. And please contact us if you have ideas of how we could make this better, or if you would like to use a tool, but you don't know quite how to interpret it or apply it. And thank you for your time, and thanks for letting us share this new resource with your audience, the folks at IEDB. We really appreciate the opportunity to work with you on this.
Excellent. Thank you so much, Betty and Will. We have a question that has come through. So if you'd like to take a moment and have a look. And there's also still a little bit of time. So Betty, I'm not sure if you want to play around with the other demos that you had, but yes, so there is a question here. If Betty or Will, you'd like to take it?
Let's see, Will, why don't you take it? I'm still trying to get back to the questions. I closed things down to try to get to the screen.
Sure. So the question is, do you have data about the spike mutation in Latin American people? And the answer, I guess, is yes. Let me go back to, I'm going to go back to my screen for a second. Hang on.
We're seeing you, not your screen. Yeah.
Yeah, thank you. So in the top 20, we have, although there are many more sequences than this from Brazil, we have roughly 1,000 sequences. Yeah, just over 1,000 sequences from Brazil, and if you look here, you can see that those are almost all either the GH or pure G form, and mostly right now what's been increasing recently is the GR form. And so that's the form with the 614 mutant, the G14. There aren't a whole lot of sequences from South America generally. The total of some 2,000 in total. So it's not representative of the full wealth of virus there.
Will, could I just make an interjection though? So one of the tools we have now is you can go down to a continent or a country, and you can request the most frequent mutations in that continent or country or even city. Just again, any geographical level in GISAID, you can break it down, and it will show you the most common mutations in spike for that country. So that tool is, I think it's available on our development site now. It should be out soon. So that will just that could tell you quickly what new mutations are present. Most of mutations are extremely rare still in spike and in the other proteins. So just these major clades, you're getting a fairly high frequency. And then there's a couple of additional mutations like the spike A22 and 477 that I used to illustrate how you might use some of our tools. But there are some of these rare mutations that are coming up, and sometimes you'll get a particular mutation that just comes up in one place. Like I was just noticing a mutation in spike that's dominated Utah. So if you have an interest in a particular country, you can focus down on it, and you can write to us afterwards and we'll help you figure out how to do that. We would be glad to. If you're curious about mutations that are coming up in your clinical site or the place that you're studying.
So I see there's another question here, which is, apart from the S protein and following the GAG protein example from HIV, is there a homolog protein in SARS-CoV-2 virus which may be crucial for virus assembly? And I think the answer is yes, the nucleocapsid, for instance. But there are a number of structural proteins as well as non-structural proteins. And point one, they haven't all been adequately defined in terms of their role. And part two, I don't know. I'm sorry, the details. So in other words, not everything is known about the structural functions of every protein in the virus, but more is known, broadly speaking, than I know myself.
Excellent. That almost brings us to time. Will, Betty, is there anything else you'd like to add in the last couple of minutes?
No, I think that's everything. But please do write to us if you have ideas and if you have questions, because we would be glad to help you play with the data and look at it from your particular context.
Excellent. Thank you both so much. We'll, of course, share your details in the follow-up email as well. So all of our attendees have your details to contact you if they would like to. And so once again, thank you, Betty. Thank you, Will, for that wonderful presentation. It's really exciting when we get to see things happening in real time, really topical, big issue for this year. So appreciate that. And now we're actually at our midway point of the webinar, of the workshop rather. And we'll be taking a bit of a longer break now, so just under 30 minutes. We feel free to step away from your computers. I think we all should have some lunch as well. And then we will reconvene at 11:45 a.m. Pacific time. So as I said, just under 30 minutes. And we'll be hearing from Bjorn when we get back, and Alex as well, and then Alba. So with that, I will thank you and enjoy your break. Once again, 11:45, and I'll pop that in the chat with some additional resources.